Hi, I'm Lee Bardwell, and this is my toolkit for system cell biology. This is video number 1 in the toolkit for system cell biology, and in this video I'm going to start to tell you about the 3 elementary reactions that we can use to model virtually any biochemical pathway with just these 3 reactions. These 3 reactions are, as you can see, the 0, first, and second order chemical reactions. And in later videos, I'll show you how you can string these elementary reactions together to build models of biochemical pathways and cellular processes, including simple models of gene regulation or enzymatic catalysis, etc. And indeed, we can make our models more and more complicated by just adding further examples of these 3 elementary reactions. So they really are the place to start. In a zero-order chemical reaction, a reactant A is created at a constant rate K. A could be a transcript that's transcribed at a constant rate. It could be a protein that's translated at a constant rate. We could use this reaction to describe catalysis by an enzyme that's completely saturated, etc., etc. This scheme makes it look like A is being created from nothing. Of course, A is not being created from nothing. It's being created from precursors that we're not bothering to include in our model. This is a chemical reaction that is zero order because the rate does not depend on the variable parameter. The variable parameter, the one that's going to change during the course of the reaction, is A. And the rate for the zero order chemical reaction is simply K. We could say instead that the rate equals K A to the 0. A to the 0 is 1, of course, which just simplifies to K. But that 0 gives you an indication of why we call it a zero-order chemical reaction. So, some nomenclature. This is called a scheme, a chemical scheme. K and A are parameters. K is a fixed parameter because the value of K is not going to change as the reaction progresses. A is a variable parameter or state variable. And we call A a variable parameter because the value of A changes as the reaction progresses. And the term state variable means that the state of the system is basically defined by the value of A. Okay, so the zero-order chemical reaction has a rate equation, dA dt equals K. What this equation is telling us is that the rate at which A changes as a function of time, dA dt, is equal to K. What we have here is a differential equation that is ordinary. It doesn't mean it's boring. It means it has no partial derivatives in it. It's first order, and that means it has only first derivatives in it, no second derivatives, etc., and it's linear. The rate of change of a chemical reaction has units of amount per time. Amount could be concentration, amount could be molecules per cell. In this case, the rate coefficient K also has units of amount per time, but we'll see that rate coefficients take on different units depending upon the situation. This rate equation can be integrated, and I won't show you how to do that here, but there's plenty of places to find that. And the exact solution that results, also known as the integrated rate equation, is as follows. A as a function of t equals A naught plus kt. Okay, what does this mean? The amount or concentration of A at any time t, that's this part here, is equal to the starting amount, A naught, plus K times t. So the rate equation, again, tells us how the rate of change of A varies as a function of time. The exact solution, or integrated rate equation, tells us how the total amount of A varies as a function of time. You get from here to here by integrating both sides. Again, I'm not going to go over how to do that. But when we see an equation like this exact solution, we can do some simple things just to make sure in our own heads that it makes sense. For example, we can plug in values of different values of t, for example, or different values of a naught, and make sure that it makes sense. So, and we can also check that the units balance. So we know that dA dt is going to be in amount per time. Here I'm using micromolar, which is a concentration per second, right? Those are my units. So if those are the units of amount per time, then K is also, as I said, has units of amount per time. Then A of T is going to have units of micromolar. It's a concentration of this chemical species. A naught also has units of micromolar. And then over here, as in our integrated rate equation, you can see that we're multiplying micromolar per second times second, right? And that works out to just be micromolar. So everything ends up being in micromolar, which makes sense. And let's look at our initial condition. When t equals 0, then A of t, according to this equation, this is just going to go to 0. A of t equals A naught. So when at time 0, you have the amount that you started with, which makes complete sense. All right, let's plot this rate equation, dA dt equals K. So we're going to plot rate on the y-axis, and rate, of course, is dA dt, and we're going to plot time on the x-axis. So rate as a function of time. Now the rate is constant at all times, so that's why I plotted a flat line here equal to the value of k. So if k is 2, then our flat line is at y equals 2. Here's a plot of the integrated rate equation, assuming that we're going to have A0 equals 0. The amount at time 0 of this species A is 0. And we're going to, again, let k equal 2, which is why the slope of this line is 2. And a plot of the integrated rate equation is a plot of amount versus time. So we have amount of A on the y-axis and the time on the x-axis again. And we can see that we start out with 0, but then the amount of A increases constantly. So after 5 minutes, we're going to have 10 units of A, right? Assuming that A0 was 0, that K was 2, and that our units are going to be in minutes, okay? Here I've plotted both the rate equation in green and the integrated rate equation in blue using Mathematica, and I'm going to start by varying the amount of A0, and that's just varying the amount we start with at time 0. Doesn't change the rate of accumulation because I've set the rate here equal to 0.4. Now let's see what happens as we vary the rate, increasing the rate. Of course, the rate goes up as we increase the rate, but also the slope of the line, that is the rate at which A accumulates, is increasing as we increase the rate. Finally, let's plot the rate of change of A versus the amount of A. In the case of the zero-order chemical reaction, the rate is independent of the amount, and so if the rate is 2, it's just a flat line independent of the amount of A. In other words, this is essentially a plot of the rate equation, dA dt equals K. Notice that the plot is a straight line, and that's why it's a linear differential equation. Before we leave this topic, let's note that we could also use a zero-order chemical reaction to describe a scheme where A is destroyed at a constant rate, or A is moved into a different compartment at a constant rate. In this case, our rate equation would be dA dt equals negative K, the negative to reflect the fact that A is going to be decreasing as time goes on. And the exact solution would be A at any time t is given by the amount you started with minus K times t. And of course, we'd have to put conditions on that so that the value of A, which we're imagining to be a protein or a transcript or some other chemical species, could not become negative, because that would be ridiculous. So to summarize, we've been talking about the zero-order chemical reaction, where A is created or destroyed or imported or exported at a constant rate. The rate of change of A in this reaction does not depend on the amount of A, and thus the rate does not change over time. We've learned that rate equations can be modeled with mathematical differential equations. We've learned about integrated rate equations, which are exact solutions to some of these rate equations. And in the case of the zero-order chemical reaction, the integrated rate equation tells us that the amount of A changes steadily, indeed linearly, over time.